Hello, this is DrPaulCottrell, and I'm going to do a video. I'm recording this on the 2nd of January, 2025. There's a virus that is spreading in China, but also in other places of the world. But there's a little bit of a concern with China with this hMPV virus that a lot of people probably don't know about. So I'm going to do an overview of this virus. It's called a Metapneumovirus, and this is going to be a series of different lectures that I'm going to do. There's a little bit of detail here. This is actually the easy part of it. And I'll be doing subsequent videos going into certain areas of detail in terms of the genome and in terms of how this virus shuts down cellular processes so it can replicate in the cell. The reason why I'm doing this is that there's going to be a series of viruses I think that we need to be up to speed on in terms of how they work and in terms of how they relate to the crisis in 2020. And those other viruses that I think we need to pay attention to is norovirus, RSV, the metapneumovirus. So I'll be doing subsequent videos on this. In addition, Barrick worked not only on beta coronaviruses, but also on uh, dengue and noroviruses. So just keep that in mind. So if you'd like to help support my work, please click the link to my website, the-studio-reykjavik.com, and help support my work. Please share the links to the videos I make. Ask your social media to follow me on all my channels. I have 4 channels on YouTube. I have Brighteon, Bitchute, and Rumble. Please follow me there. And please help support my work by donating. You can donate through Stripe, PayPal, or buy me a coffee on my website, the-studio-reykjavik.com. All the links are in the description of this video. You can also be a paid member to my Patreon channel, and that way you're helping support me producing videos like this and covering the news. I have thousands of videos for free, so it's through you going to the website, purchasing the products that I have, and donating that helps me produce these videos. All right. With that said, we're going to start. So again, like I said, there is this human metapneumovirus. All right. There's also an avian version of this, an avian MPV. So there's a human one, hMPV, and there's an aMPV. All right. This is a relatively new virus that was discovered in 2001. The avian version of it was discovered in 1970s. All right. They think through genomic analysis and lineage and some statistical mathematics uh, that there was a split between human MPV and aMPV about 133 years ago. So this is a relatively new virus. All right. I'm just concerned because there seems to be a lot of NIH activity for funding in 2003. And it's very close to RSV. I mean, it's different, but I mean, there's a lot of similarities. So we know that there were nefarious activities going on with the national security state through biological defense, going all the way to pre 9/11. And the funding that went to Barrick for uh, investigating beta coronaviruses that eventually led to the SARS platform uh, started in April of 2001 for the funding. And then, eventually the first SARS virus was detected in China in 2002. So it's important to, keep in mind these data points. So hMPV is a relatively new discovered virus, and even with Bayesian mathematics and modeling avian MPV, that that that's the split, the divergence seems to have happened in a relatively short time period. They're predicting approximately 133 years. So if that's correct, if the information that I read was correct, there's something's weird about this. I get this feeling that there's something not right with this virus. All right. So that's the reason why I'm doing this video, to find out, why do I have this feeling? All right. So this now we're going to get into the details. All right. So hMPV has a lot of different proteins. All right. And what's shown on the screen is the structure of the virus. It's a little bit more complicated in a way compared to what we've been seeing with other viruses that we talked about, especially the beta coronaviruses. So let's go through this structure here. Let's start with the outside and go inward. So you have these different proteins, right? You have a glycoprotein, which is just like your spike that attaches to a receptor. And you have a fusion protein that helps with fusing to the cell. You also have a biolayer, a lipid biolayer, a lipid bilayer there. And so that's nothing special. Underneath that, you're going to have a matrix protein, right? And you also have something called a SH protein. We're going to go into detail what all these are. You also have a polymerase. Now we're moving inward here. You have a polymerase that is attached to or on the negative sensed RNA genome. And then you also have phosphoproteins on your RNA. And wrapped around that RNA is these nucleoproteins. Okay? So you have fusion proteins, help to fuse to the cell, that allow for cell fusion. You have glycoproteins, those are the spikes that attach to the cell. You have a matrix, you have a lipid bilayer, you have the SH protein, you have a negatively sensed RNA, you have phosphoproteins on that negatively sensed RNA, nucleoproteins that package the RNA, that kind of protect it. You have a polymerase, which is important. All right, so let's go to the next slide here to go into what does the genome look like. All right, so the genome on the right codes for these different proteins that I was talking about. So again, the nucleoprotein is the N. And what does that do? Well, what I'm going to do is instead of going and saying what does it do, I'm going to just go over what the genomic structure is first. So at the very beginning, at the three prime part of the genome, right? The beginning of the genome, you have the N protein. Okay, that's the nucleoprotein. Then you have the P, which is the phosphoprotein. Then the M, which is the matrix. Then you have a fusion protein, which we talked about. Then you also have additional matrix proteins called M2. And you have two types. You have M21 and M22. And we'll go into the details of that. Then you have you have this SH protein, right? This is the small hydrophobic protein. And then the glycoprotein, this is the spike. And then the large polymerase protein that allows for the genome to be replicated. All right. So we already talked about where the proteins are through the virus structure. Now let's talk about what does the N protein do. So basically the N protein, like I mentioned before, is that it kind of wraps around the genome, the RNA genome of this virus. The RNA genome of this virus is a negatively sensed RNA. Beta coronaviruses are positively sensed RNA. So this is a little different. Okay. We'll go into details about that. Phosphoprotein is a cofactor for the polymerase that allows for replication, translation. Matrix protein, this is for the viral assembly and budding. Fusion protein, this is a virus host cell binding. All right. So this is how the virus actually binds to the cell surface. Right. And it has a unique feature I'm going to be talking about in a subsequent slide. The M2 protein, again, has an M21 and an M22. So there's two parts to this M2 protein. The M21, right? This is a matrix protein. The M21 is the RNA transcription. Okay. The M22 balanced transcription and genome replication. So again, because this is a negatively sensed RNA, it has to go through a polymerase, right? That allows it has to go through a transcription that allows for the negatively sensed RNA to turn into positively sensed messenger RNA. And then the proteins of the virus can take hold. So there's a mechanism to turn the negatively sensed RNA into positively sensed messenger RNA. And that's the RNA transcription. And then the M2 type helps to balance that transcription and then also genome replication. Now, to replicate the genome, what's going to happen is that you have to convert the RNA negatively sensed into a positively sensed RNA. That's messenger RNA that leads to that's capped and it has the alpha. It has the poly A tail. All right, so you have messenger RNA that's capped with the poly A tail that's positively sensed. It'll create proteins that are needed for the virus to assemble and to replicate. You also have this positively now sensed RNA that's created. And through replication complex, it'll take that positively sensed RNA and make a complement or a negatively sensed RNA that will eventually get packaged for the virus. It'll get coated with this nuclear protein, this nucleoprotein, and get packaged with the polymerase and some other proteins. Like for example, we were talking about the phosphoprotein will get packaged with that negatively sensed RNA that's been replicated. You'll get packaged with the nucleoprotein, and then you'll get some polymerase that's packaged with it. Okay. So that's what's all right. So now the SH protein, the small hydrophobic protein, that's not it's water fearing, hydrophobic. All right. So this virus is relatively new, so there's not a whole lot known about it. There's probably more known about certain aspects of this virus is probably more known than let's say Mpox. Mpox is a really complex virus that has a lot of different proteins in it, and it's a more complex protein. I don't think that we know that much about pox viruses, but we don't know much about MPVs either. All right, so there seems to be some evidence to suggest that there's a viral poron. And that is basically kind of a channel that allows for the cell to bud out, to kind of be released from the cell, the virus from the cell. And we'll go into how it inhibits the immune system. There seems to be some good evidence on that. Now, glycoprotein, this is the spike, all right, and this is the virus host cell attachment. All right, so just that glycoprotein is how it targets, but it uses the fusion protein to fuse, right, with the cell. And then the large polymerase protein helps to do the viral replication. All right, and there's going to be some of these proteins kind of bind in a complex to help make the negatively sensed RNA. Now, let's talk about the hMPV replication cycle. Okay. Now, when we look at this diagram here, we're just going to do another review because this is a little different virus. All right. Okay. You have a negatively sensed RNA genome that's here, and that's wrapped with nucleoproteins. It's wrapped with phosphoproteins. It's also wrapped with M2s, M2 proteins. And you can see that you have an M1 and M21 and M22. And then you have a large polymerase. And that's inside the virus. Now, in terms of the matrix, you have a matrix, right? You also have a lipid bilayer, but it's not shown in the diagram very well. Now, and you also have the glycoprotein, that's the spike. You have the small hydrophobic protein that might be a viral pore to allow for the virus to get out of the cell. And you also have the fusion protein. Okay, so that's the structure. Now, how does it kind of replicate and how does it translate? Right? So there's transcription and there's translation and then there's replication. The transcription is when you are taking the negatively sensed, so this virus gets into the cell and it starts to do its thing, right? There's a release of the negatively sensed RNA into the cytoplasm. And what is happening is there's these inclusion bodies that are created to help with the virus to replicate. Okay. So, but we'll get into that a little bit later. But basically, the negatively sensed RNA gets to released out of the virus package and into the cytoplasm, all right? And then their inclusion bodies are involved. But as this viral negatively sensed RNA can't create directly proteins, it needs to go through a transcription, all right? And then a translation. So to create code, RNA code, DNA code, either from DNA to RNA or RNA negatively sensed to RNA positively sensed, that's transcription. So it's something called a primary transcription. So it's taking that negatively sensed RNA and it's doing a primary transcription and creating positively sensed messenger RNA. That's positively sensed messenger RNA is going to be capped and then poly-A-tailed to allow it to create proteins. So it uses ribosomes, right, to create proteins through translation. So you have transcription from negatively sensed RNA to positively sensed messenger RNA to a translation that creates the protein, okay, with ribosomes. So, and it's using the cellular ribosomes to do that. Now, the proteins, the viral proteins here that are created from this positively sensed messenger RNA, these proteins are the spike, the fusion protein, the SH protein, the matrix, right? The M2 proteins, right? And the large polymerase, the nuclear protein, and the phosphoprotein. Now, when these proteins are created, some area of the cell starts packaging the viron in certain area. These self-assemble and it's being packaged in the cell, and the Golgi apparatus helps to create the viral particle. All right. So these proteins are starting to build the virus structure. All right. But you also have these three proteins, right? And that's the polymerase, the large polymerase. You have the nuclear protein and the phosphoprotein that will bind. They're a complex. And this complex helps to replicate the genome. So the first step is to create the proteins through converting negatively sensed RNA into positively sensed messenger RNA, create proteins. These three proteins, the large polymerase, the phosphoprotein and the nucleoprotein bind, and they help to create the negatively sensed RNA genome that will get packaged eventually with these proteins that were created earlier. Okay. Now to do that, you can't just replicate a negatively sensed RNA. This negatively sensed RNA needs to be the complement. It has to be a positively sensed RNA to be able to replicate off of it. So this complex turns a negatively sensed RNA sequence into positively sensed RNA that goes through an additional process of taking that positively sensed RNA, viral RNA genome and make lots of the opposite. So this complex is turning this into positively sensed RNA that won't work in the virus, but that is a template to create the negatively sensed multiple copies of RNA. And so what happens is it, creates a positively sensed one first, which is called an antigenome, and then it will create a negatively sensed RNA and create this complex. Okay. And that complex has the nuclear protein on it and the nucleoprotein on it, the phosphoprotein, and the large polymerase. So the key here is that you create the you 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 have you have you have additional proteins that are created to help the replication process of the genome for the virus. All right. You also, to start this in motion in low yield, you have the 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 packaged polymerase to help with this early transcription. All right. So you have this early transcription that's taking place. Right. You with these breaking off, you you might even have early low yield, negatively sensed replication happening in the cell. But when the proteins are more and more proteins are being created in the cell based on this this positively sensed messenger RNA, right? Over time, you're going to create lots of viral proteins, including the replicase complex. And once that's created, then lots of negatively a negative genome or positively sensed RNA are created that will eventually be reversed into negatively sensed RNA. So that's how it works. Okay. A little bit different than positively sensed genomes for viruses like beta coronavirus. Because in beta coronavirus, if you have a positively sensed RNA, you can directly go right into making proteins. And then if it's positively sensed code, then you have to reverse it and makes a negatively sensed code as a template to make positively sensed. But in this case, it's the opposite. It has to take this replication complex. The hMPV has to take this replication complex to help replicate the genome off of the negatively sensed RNA, make it into positively sensed RNA, then that's a template to make lots of copies of negatively sensed RNA. And the key is that there's this wrapping that's taking place with the polymerase, the nucleoprotein, and the phosphoprotein to protect the RNA. Okay. So that's all long-winded on how it replicates in the cell. Now, how does it inhibit cellular processes? Cells have certain defense mechanisms, but these proteins shut them down. Okay, so let's walk through. This is a little complicated slide. And I'll go into maybe a little bit more detail of each one of these pathways in a subsequent video. So the human metapneumovirus, all right, the human metapneumovirus gets into the cell. All right, how does it get into the cell? First, the spike protein, the G protein, attaches, and then the fusion protein helps to get it into the cell. Then there's a release of the virus of this single-stranded, negatively sensed RNA. This RNA, okay, would be detected by the cell normally. Viral RNA could be detected by innate pathways within the cell to activate the cell to go into antiviral mode and to kill off the cell. But this virus has some inhibiting functions. So as this viral RNA is in the cytoplasm, okay, it can be it'll attach to something called RIG1. This is kind of an innate immune system kind of mechanism within cells. So you got RIG1 and this MDA5. All right. And I'll go into details in a further video because there's different cells that activate different things here. But the two big ones are the RIG1 and the the MDA5. Now, this viral protein, or not viral protein, this viral RNA is going to go through this process of making proteins, right? Like we said here, it's going to make lots of proteins. So it's going to go through a transcription, create messenger, positively sensed messenger RNA, and then create lots of proteins. So lots of proteins here that are being created in the cytoplasm, and they can shut down certain things. So this pathway, okay, normally a viral RNA activates RIG1 or MDA5 and then activates MAVS. Okay? And MAVS activates NF kappa beta. And then that signals that it gets phosphorylated, and then that signal gets into the cell and goes into a pro-inflammatory cytokine creation. All right, it tells the cell, "Hey, I'm infected." Okay, that's how normal viruses activate the innate immune system within a cell. It comes in, it gets detected by MDA5 or RIG1, and then it goes to MAVS, which is on the mitochondria, and then all of a sudden you have NF kappa beta pathway phosphorylated, and then boom, activate pro-inflammatory cytokines. Okay, it goes into antiviral mode. Okay, another pathway that is activated in other viruses is once MAVS is activated, not only is it going through the mitochondria and activating NF kappa beta, but it's also activating IRFS. And then IFRS is also phosphorylated. I'll go into detail on each one of these pathways. I just don't have time in this video to do it. And then the IRF3 phosphorylation will get into the nucleus of the cell and then go into to help create the interferon beta and other interferons type 1. Okay, so you have interferons being created by the IRF3 pathway and the NF kappa beta pathway. It's pro-inflammatory cytokines and type 3 interferons. Okay, but notice you have to have a phosphorylation to take place for it to get into the nucleus for signaling. Another innate property for cells in normal, let's say, viral infection is that you have TLR activation. Okay, what happens is that the viral genome is detected, all right, and it can be detected by TLR7 or TLR3. And if it's detected by TLR3, then we'll use this TRIF pathway and then it will use the IRF3, not the the IRF3 being activated by MAVS. Well, IRF3 can also be activated by the toll-like receptors, number 3, through the TRIF pathway. All right, gets phosphorylated, comes into the nucleus, and again, interferon beta and other type 1 interferons are created. So it signals the cell, hey, I'm infected. Okay. Another pathway with typical virus infection in a cell is through the TLR7, and that will activate MyD88, and that pathway is through IRF7. And then that gets phosphorylated, comes in, and then produces interferon beta and other interferon type 1s. Okay. Okay. In addition, IRF7 in dendritic cells will activate NF-kappa beta, allow for phosphorylation, and then go in a pro-inflammatory cytokine profile and type 3 interferon. Okay, now we have, now we're going to go into how specifically hMPV can shut down these pathways. Again, in normal situation, if a virus can't do the shutdown of the innate immune system in the cell, you're going to get RIG1 or MDA5 activation. You'll get the MAVS activation. You'll get the IRF3 activation, and eventually these a antiviral activation and to try to kill off the cell and tell other cells to go in antiviral mode. You'll also have the NF-kappa beta pathway activated. You'll get these toll-like receptors activated, and you also get the IRF3 pathway and the IRF7 pathway activated. Okay? Now, this virus, hMPV, has special capabilities. All right? As the virus genome gets released into the cytoplasm and gets primary transcribed, all right, that primary transcription, you're going to get the messenger RNA positively sensed and create lots of proteins. And lots of those proteins that are being created, all right, that's this viral messenger RNA then there's this messenger RNA that's positively sensed gets translated into proteins. As it's creating proteins, lots of proteins, all right, this viral messenger RNA translation looks to shut down the interferon alpha receptor and JAK1 and TYK2 pathway and stop the signal for the cell to produce that. Okay, so now, but there's a little bit of like exactly how that happens is still left for research. So in the early translation of the messenger RNA that's positively sensed, there's a shutdown of interferon alpha. Now, as these proteins are starting to be made, and we talked, there's the P protein, the G protein, and the SH protein, right? All these different proteins that are being created in the cytoplasm, they're shutting off these different mechanisms. For example, the phosphoprotein P, all right, that will shut down RIG1 and MDA5. When G protein is created, which is the spike, that will shut down the pathway for RIG1 or MDA5 to activate MAV1 or MAVS. All right. The SH protein, that helps to shut down the NF-kappa beta pathway. which is major pathway for stealth in the cell for a virus. Okay. Now let's go to the M. Remember, I was talking about the M2. So you have the M22, which is also up here. The M22 protein can shut down the signaling from the TLR7. All right. That means that it's inhibiting MyD88 and then preventing the IRF7 to activating NF kappa beta and not being allowed to be phosphorylated and preventing the cell from creating interferon beta and other interferon type 1s. Okay. So M22 will help to shut down the TLR7 pathway. There doesn't seem to be a known protein that shuts down the TLR3 pathway. Now, so that's really kind of important. Now, there's seems to be some questioning here that, okay, you have the G protein interacting with the TLR4 that activates MyD88, which probably will activate the IRF7 pathway if it's in DC cells, dendritic cells, but I'm not sure if it's non-dendritic what that pathway is. I'd have to research that a little bit. But the point here is that there's lots of proteins that the hMPV produces that shuts down the innate immune system in the cell and prevents the cell to going into antiviral mode. It prevents the cell from telling other cells, go in antiviral mode. It prevents other cells and this cell to warn the immune system it's infected to kill it. So so it allows for the virus to spread. So the reason why I'm going over hMPVs is that many of the proteins have inhibitory functions that are in that will shut down the innate immune system in a cell. Now, you take this, another reason why I'm doing this video, not just to go over the details of this virus, but also how it couples with the shutting down of the innate immune system, the weakening of the innate immune system, and the weakening of our adaptive immune system since the crisis in 2020. So if you already have a weakened immune system, then you're going to be more susceptible to hMPV. And this is why I've been saying for 5 years that AIDS-like syndrome is going to be very, very problematic with other pathogens that are either viral, bacterial, or fungal. And this is a classic example where you have a weakened immune system and it doesn't take much for this to take hold. Okay, so that's a long-winded explanation on how the innate immune system is shut down. Now, what I want to transition to is how the genomic similarities between RSV and human MPV are. Now, remember, I stated that there are two types of MPV. There's avian MPV and there's human MPV. Avian MPV was discovered in 1970s. Human MPV was discovered in 2001. Statistical, mathematical, Bayesian mathematics is, predicts, and I'm not sure how valid it is, but predicts that the split between avian and human MPV was 133 years ago. Not long ago. All right. So this is a relatively new virus. All right. And it's during, human exploration in biotech. All right. You know, 133 years ago, our biotech was pretty naive. It's still naive. It was in a nascent form. And is it possible that we were doing things in the laboratory that just, mixing stuff and creating inoculations that led to the formation of this? I don't know. I don't know. I'm not going to speculate. I just want to just state the facts. But this is a relatively new virus that's been discovered, and the Bayesian analysis of the actual lineage of the split from avian to human was about 133 years ago. Okay, could be a little bit less. Now, how accurate that modeling is, who knows? But just keep that in mind. So you have this hMPV that's relatively new, probably split off of avian MPV according to Bayesian mathematics, about 133 years ago. Now, this genome, like I said, has an N. So you have your nucleoprotein, you have your um, phosphoprotein, you have your matrix, you have your fusion protein, you have your M2, 1 and 2 protein, you have your glycoprotein, you have your before the glycoprotein, you have your SH protein and the glycoprotein, and then the um, the polymerase. Okay, so that's the genome. Now, there's a similarity between RSV and hMPV, and not just hMPV, but also avian MPV. All right, and that is one, they are similar in length. RSV is a little larger by around 2,000 nucleotides. Uh, so the genome size is about 13,330. RSV is 15,222, roughly. All right. So with that said, you have you have you have this sequence for the genome. Now, what are the differences between human MPV and RSV? Okay. Well, it's very, very similar. The difference is that the hMPV doesn't have non-structural 1 and 2 proteins that RSV has. So it's as if hMPV lost something. Okay. Now, of course, one has to dive in and look at the differences between the N protein for RSV, the P protein for RSV. How much homology is there? I don't know. I suspect there's quite a bit. All right. So with that said, you also have your nucleoprotein, your phosphoprotein, your matrix. You have your SH protein. How much of homology is these? It has to be investigated. You also have the fusion protein and the glycoprotein. It'd be interesting to know if these glycoproteins are similar. You also have the M2. All right, but so it's like rearranged. I'm just showing similarities. I'm not saying they are the same and that they're recombinants of each other. That's not what I'm saying. I'm just saying that the genome structure is similar. All right. Now, big difference is RSV has NS1 and NS2, non-structural 1 and 2 proteins. Now, another big difference is the arrangement. All right. SH protein in the human MPV is after the M2 section of the genome, while in RSV it's in between the matrix and the glycoprotein. All right. Another big difference is glycoprotein is before the fusion protein in RSV and not in hMPV. So those are, some of the interesting features for RSV. It's not the only. I would be curious to understand metapneumovirus homology to orthopneumovirus for these different proteins. What's the percentage of homology? I suspect that it's pretty high, but I don't know. I haven't checked. But there's a similarity between RSV and hMPV. Now, hMPV currently in China is causing pediatric issues with patients that are younger than 14 years of age, and patients that are 65 years and older, very similar to RSV. So even clinical manifestations are similar. Okay. Now, the last slide, I want to explain how other cells can be infected. Now, we went over how the innate immune system is shut down by the different proteins that the hMPV create. So it shuts down the innate immune system and spreads. It can spread two main different ways. And the second way has three subtypes. The first way is the normal exocytose budding off, right? You have budding off, you can have budding off or exocytose viruses from the cell just getting into the interstitium, into the matrix between cells. And then the glycoprotein attaches to another cell's receptor, and then it gets in through the fusion protein. That's kind of like the standard model for viruses spreading from one cell to another. It's being created, right? A part of the viral packaging is, moving up to the cell surface. You have you have the negatively sensed RNA package. It wraps in with the viron shell, and then it then buds off, right? And that's why they were thinking that maybe the SH protein helps with that budding off process. In addition, you can have viral loads so large in the cell that the cell kind of just bursts, right? All right, so you have the budding off thing, but you can also, because of the fusion protein on this hMPV, it can help bind other cells to each other. Okay, so just imagine, this graph doesn't show it very well, but just imagine you have this part of the virus at the surface of the cell before the negatively sensed RNA is packaged in. So you have kind of like all these, you have the SH protein, the fusion protein, the glycoprotein at the surface of the cell, and it's starting to bud out, but it isn't fully packaged yet. And then you have the shepherding of the negatively sensed genome coming in, and this thing's about to bud off. Well, you have a fusion protein here that could help, and even the glycoprotein help attach to another cell that's adjacent to it to kind of allow it to stick. So you get these conduits that get created. All right. And there is something called actin within cells that help to transport this. All right. So you have the potentiality of cells touching each other because of the fusion protein and the glycoprotein. primarily the fusion protein. So they can like they can allow for fusion of cells. Okay. But you also can have these conduits that are taking place with these cells that are like attached to each other now. And these conduits, right, can pass on fully formed particles, viral particles, and follow along the actin chain. Okay. So there's three ways that this can spread. In the second subtype. The first subtype is budding off. The second subtype is through this channeling fusion of the membranes of the cell. So there's three different versions of the second type. The first is that the particles spread from cell to cell across open-ended extensions. Okay, so the cells are bound together, maybe from the fusion protein and the glycoprotein that was at the cell surface that allows for this to connect and creates a channel. And these channels, though, and there's other reasons for these channels to take place too. All right. So these channels could be open-ended and, allow for communication to take place. All right. Sometimes cells stick to each other and they have pores, and these pores allow for cellular communication to take place. And this virus may be using one of those pores as a method. That's another possibility. But again, there's you know there's research that has gone in on MPV, but it's a relatively new virus, and there's some things that are a little bit unknown at the moment. All right, so you have this transfer of the virus along the actin and going through the channel. You can also have a movement of the virus particles on the outside of the intercellular extension, and it's just kind of following along, and then it attaches. Okay, so you could have it budding out and attach. You can have a channel that's been created by some method, either the normal channels that are created between cells, desmones and all this stuff, and there's these pores that can be created in certain tissues. But you also could have a series of fusion proteins or glycoproteins on the cell surface from the virus that help with the fusing of these two. And then you have this actin movement. So it can be following in the channel, it can fall outside and just kind of roll and then eventually fuse, because maybe this channel's too small to actually fully go in, and therefore it has to kind of just roll around until it gets to the larger part of the cell. Then there is another way to do it, and it's a spread of replication bodies. All right. And you can have these replication bodies where it's not fully packaged. Okay. But if you look at this, if you paid attention to the slide that I was showing here, right? This is your RNP complex, negatively sensed RNA, as it gets packaged into the virus, the virus structure, the shell of the virus, right? So this is the NP. But as this is being formed, there's an inclusion body that's kind of helping this along. Well, that inclusion body or replication body, which isn't fully packaged with the cell, could travel along the pathway through actin and have it spread between cell to cell. So you can have intercellular movement, and some pathogens do this, all right? Intercellular movement between cells, the budding, right? Or a rolling effect that can be happening. And it can be either through an inclusion body, or it can be through a fully packaged virus going through the channel, fully packaged virus rolling on the surface of the channel to the other cell, or budding off and then connecting. So those are the four different methods, roughly, on how it can spread from one cell to another. Now, here's what's important. Because I showed you how the innate immune system can be shut down in an infected cell, you can, and shutting down, telling other cells around it, adjacent cells, shutting down that antiviral capability. In a normal situation, in a situation where the innate immune system in the cell is working 100%, if a virus hits the cell, it will tell the cell to create all these pro-inflammatory signals, tell other cells to go into antiviral mode and to die, and that if infected, die off quickly. And this cell, after it signals other cells, it will try to die, go through an apoptosis. Okay? But if you have viruses that shut down these pathways, then the cells can't communicate very well to adjacent cells to go into antiviral, so the virus spreads. And if the shutdown's adequate, this cell doesn't go into apoptosis. All right. So this cell's infected. All right. And this cell's innate immune system is shut down. So it can't tell this cell to go into antiviral mode because all these pro-inflammatory mechanisms have been shut down, because these things get released from the cell, and these pro-inflammatory cytokines signal, and especially the NF-kappa, alpha, and beta, signal to other cells go into antiviral mode. Well, that's not happening because the innate immune system's been shut down. So when that happens, it buds off and it can attach, and this cell thought everything was okay. But when you have a channel coming on here, not only do you have a channel of the virus moving around, you also have a channel of proteins moving in. And these proteins, as you know, like G protein, can shut off RIG1. So if G protein gets through this channel and comes through, all right, it has already started in motion shutting down the innate immune system pathway through this channel. So that's another problem with intercellular transmission, through channels with pathogens, because these channels not only move the inclusion body, move the replication body, but can also move viral particles, pieces of viral particles, can roll the viral particle on the outside, but also transfer proteins through the channel into connecting cells, shutting down their innate immune system. All right. So that's another key feature to kind of pay attention to. So that's the overall structure and capability of human MPV. Now, again, this slide is really important to understand the shutting down mechanism. I don't have enough time in this slide in this video or this slide to really go into the details of what exactly each of these pathways are. I will do that in a subsequent video, but I have mentioned and how how certain inoculations can shut down pathways, shut down the innate immune system, and prevent the cells to go in antiviral mode. And that's the whole point of the messenger RNA platform used for inoculation. So, okay. With that said, please help support my work by going to my website, the-studio-reykjavik.com. It's the-studio-reykjavik.com. The link is in the description of this video and all my videos. Please go to that link, help support my work by purchasing the products that I have and donating. You can donate through Stripe, PayPal, or Buy Me a Coffee. You can also be a paid member to Patreon. All the links are in the description of this video. And please tell your social network to follow me. Follow me on YouTube. I have 4 channels on YouTube. I have Brighteon, Bitchute, and Rumble. So I can reach more people to help navigate through this crisis. Thank you for listening. And this is the first installment of the overview of Metapneumovirus. Thank you for listening and have a nice day.